This method can answer key questions in the growth and morphology of stolons and rhizomes of different crops, such as turfgrasses, meadows, and pastures.
The main advantage of this technique is that stolon and rhizome length can be measured in samples with a large amount of biomass, for which weight is the only parameter currently used.
Collect samples using a soil core sampler. Include the above-ground biomass and a soil layer with an appropriate depth, depending on species, to ensure collection of both stolons and rhizomes. Alternatively, define the surface area to collect with a frame, and collect the samples with a spade. Label each sample with laboratory tape. To clean the biomass samples, place the sample in a large sieve with 0.5 to 1.5 mm openings depending on stolon or rhizome size. The openings should be small enough to retain all stolons and rhizomes, but large enough to allow the soil particles to be removed. Clean the samples with a stream of water with enough power to remove soil particles without damaging the plants. Retrieve the cleaned samples and place them in a tray with paper towels, taking care to label the trays appropriately. Further clean the samples by removing roots and leaves with scissors. During this process, separate the stolons and rhizomes, if needed, and record additional information, such as the number of plants, tillers, and stolons per plant. Place the stolons and rhizomes in paper-labeled bags,